Thank you very much for the kind introduction and especially for the invitation to speak to this so qualified audience. What I'm going to do is trying to show in this half hour what breeders have been doing for millennia of development of better varieties. And this, I make the point that this is actually maybe the main pillar of for the sustainability of modern society, or life as we know it, because all of us rely on a pact for our life that's our food supply. Actually, I think that in the United States, 98 or 99% of the people never did agriculture actually, but they don't worry about it. They know there will be food on the table. And that is only true because there is a pact based on which they will give something to society in exchange of the food they get on the table. That will be only sustainable if the varieties will yield enough food for everyone. So what breeders do is normally to work within the peak of the pyramid of genetic diversity, what we call the improved germplasm or improved cultivars. Sometimes that diversity is not enough and you want to go beyond that and look for some resistance gene or special quality gene. And so in that case, you may want to go to a broader diversity. That would be the Landraces and sometimes even the wild relatives. But that's increasingly difficult to go to really tap the genetic resources that are more exotic and less adapted. Besides that, breeders can introduce novel variation through induced mutations that had just been cited here. And that's a totally random process where you really submit DNA to some harsh conditions, either chemically or physically, and that will result in changes in genome sequence that in the great majority of the times will be unfavorable and disabling, but may in some rare cases result in something useful. And now there is also more recently the option of genetically engineering plants for specific purposes. So you could introduce carefully one gene there in the genome to get a specific result. There is still in the current methodology some randomness, especially in the place of insertion of the genes. But even for this, there's been some significant progress in science, so there may be better control of this in the future. Didactically, we can split plant breeding in 3 different eras or great phases. The first one was what Sherry just talked about, plant breeding based on observed variation, the variation that was there and then men and women selected on it. The second, and this corresponds to the dawn of agriculture about 10,000 years ago until the end of the 19th century. The second phase, which corresponds approximately to the 20th century, is plant breeding based on controlled mating, all what we associate to conventional breeding. And now, roughly from the turn of this century, we became able to actually monitor what's going on inside of the genomes. So we can now really understand the recombination within the genome. We cannot control it, but at least we can monitor it. And within that first era of plant breeding based on observed variation, we can still have 3 subphases. Plant domestication, that's taking wild plants and converting them into crops. Intuitive farmer selection, that would be the development of specific landraces for specific situations and environments. And the last one, which is in the frontier between this unknowingly breeding and the scientific breeding, that's pure line selection and mass selection, what would originate the first so-called cultivars. So let's talk a little bit about each one of those phases. Plant domestication, or the origin of crops. That has been very well explored by Sherry. But I just want to make or reinforce some point here, that it has been actually one of the pillars of the foundation of civilization itself. We wouldn't be here under this roof today without the domestication of the plants. We would probably be roaming and gathering something to eat instead of this. And a disability in nature is actually an advantage in agriculture. So we can see it in a very interesting way. You can imagine that a disabled plant met a hungry person, and that person actually used the disabled plant and protected it and perpetuated it in a very competitive crop. So actually many of those plants became disabled and got extinct. Of course, the natural selection got rid of them. But some of those mutations actually were lucky enough to find a person to rescue it and to multiply it into crops. And now they became actually the dominant type. If you compare today how much cultivated rice and how much wild rice there is, actually the disabled became the dominant type. The domesticated, and this process was a tight bottleneck for genetic variability, of course. And today, what we have actually is a gene flow in the reverse direction, from the cultivated types to the wild types. And this is also one issue of discussion about the GMO safety, especially for the environment. Okay. Once we have something croppable, so started diversifying in the hands of people all over the world according to their preferences, the place where they lived, the climate, the soil, the pests, and also the cuisine. That's very interesting because people learn to like what they have. Do Japanese like sticky rice because they once decided that they like, or because the only rice they had was sticky? Then they developed the whole cuisine towards the sticky rice, and so on. So there is a co-evolution there of the grain type and the cuisine itself. And those very diverse populations, since they are diverse, they are able to evolve according to the changing pests and management and soil. They could migrate with people and become different land races. And this is actually it mimics very nicely the Darwinian laws of diversification. And their yield is stable but moderate. But it really served people for many centuries. And land races are considered highly valuable genetic resources for plant breeding. So what people do normally, go to traditional farmers and collect seeds from land races, put them in the fridge, right? Conserving them in gene banks. We do characterizing of it. We do screening of it, looking for something rare and different. So we are looking for off-types when we talk about qualitative traits, and outliers, points out of the curve when we are talking about quantitative traits. And once we found those new resources, we can incorporate it into breeding and develop new and better varieties. The sad point of this is that those new varieties will normally go back and replace the Landraces that they benefited from. So that's why it's so important to still have the Landraces in the fridge, or else we don't have them anymore. All right, and the pure line selection, that's what can be considered as first plant breeding method in the narrow sense, is going into those Landraces and picking the best individuals and open pure progenies. And when we're dealing with a self-pollinating species, normally we will get a quite pure progeny because those plants are pure lines. It's just a mixture of pure lines in that population. And when you compare them, of course, some of them will be better than the others. And then you could release that as a new variety, which will, of course, it will outperform, out-yield the Landrace from where it came from. So this is a very simple breeding method that anyone could do if we have a Landrace on hand. And here, so we are taking genetic diversity from within populations and breaking it between varieties, between different varieties. And then agriculture became more and more based on homogeneity. When we're talking about cross-pollinating species, then it's not so simple, because even if you take the seeds from a single maize plant, it received pollen from many other plants. It's highly heterozygous, so it will segregate. But normally, if you select the top performing plants, you will have a better population, agronomically speaking. And then you will have some genetic gain due to this selection. And recombination will occur naturally in the field. Okay, so concluding this first part, that's breeding based on observed variation, even if we don't have a clue about the origin or the basis of this variation. I want to put a premise that I think everybody will agree, that plant domestication and intuitive selection worked on pre-existing variation, resulting in deep changes in plant phenotypes. That's well known. And I want to make a proposition that within Landrace selection led to the current paradigm of homogeneity in agriculture. And this paradigm of homogeneity has some side effects that are well known. That less we don't have within cultivar adaptability. So cultivars do not evolve anymore. Cultivars are terminal because they are pure, thoroughly homogeneous. You have a higher vulnerability both to biotic and abiotic stresses. So it's an environment. A modern crop is something unsustainable by nature. So you have to keep that way by artificial methods, normally involving chemical inputs, because the principle of the entropy everything will tend to the chaos. So keeping things extremely organized takes energy. But the question is, could we recover agriculture diversity and thus its robustness without having a setback in productivity? So that's a challenge that maybe with the modern tools we could address. Could we do genomic selection for introducing a hidden variation that would not appear in the field? Could we produce two maize plants that would be phenotypically identical but still genetically different? If we could, that would be nice, because we could have the benefits of diversity and the benefits of phenotypic homogeneity. Okay, so moving to the second phase, plant breeding based on controlled mating, which is the plant breeding itself, or conventional plant breeding. Here we take two plants that have complementary qualities and virtues, cross them, and try to find a progeny, a sibling that would have both the qualities of one parent and the other. Sounds easy, but it's actually quite challenging because those traits are controlled by so many genes. And we can, again, didactically classify into pedigree breeding, ideotype breeding, population breeding, and hybrid breeding. So let's see here. Some critical factors for the success of a conventional breeding program. I put this list from my mind, and then coincidentally came a survey from that Plant Breeding News organized by Cornell and supported by the FAO, probably money, well, some of you may have known about this. And they matched quite well. A plant breeding program must have a clear definition of the target environments and the priority traits. You cannot solve all of the problems at once. There must be access to germplasm, a reliable crossing procedure, an efficient crossing procedure, fast generation advancement, the capacity to evaluate a large number of genotypes, because you have to evaluate a lot to apply strong selection pressure. Otherwise, if you evaluate just a few, you can't discard, and without discarding stuff, you don't make genetic progress. And you have to have representative multiple location trials representing the true conditions of cultivation and an efficient production of foundation seed. All of this is necessary for a breeding program to make an impact in the real world. The pedigree breeding is the most classical method for self-pollinating species. Here we just cross two plants and take the progenies, and in every generation we select the best plants and open lines which are increasingly pure, increasingly homozygous, and but also they are increasingly homogeneous. We lose a lot of genetic diversity in each of the generations. For this reason, when we get to the later generations, when you can run your yield trials, because for this you need a bulkier amount of seeds and so on, when you get to this point, you may not have enough genetic variance to apply strong selection for yield. For this reason, the pedigree breeding is considered less efficient for causing yield gains. But what's nice is that you can really know all the relationship between the varieties. You can even build those nice genealogical trees connecting Brazilian rice varieties with American rice varieties, with material from the International Rice Research Institute, CIT, and so on. So it's something that it's a nice system from the breeder point of view. it's just limited from the quantitative genetics point of view. So the power for grain yield gain is a little limited. But you see those nice contrasts like this for plant architecture, drought tolerance, environmental adaptation that is very motivating for the breeder. The multiple environment trials is something that every breeding program has to have, because as you go from earlier generations to the later trials, you normally test fewer lines in more environments. And then it goes increasing until a point where you really have it tested in many, many different situations until you can decide for the release of one or two of those varieties. So normally, it's a multiple step procedure that allow you to detect the problems with the breeding lines. And plant breeding is dubbed a numbers game because of the almost endless combinations that you could have of gene alleles. For this reason, the large breeding programs, they are really high throughput in seed handling, phenotyping, everything. Ideotype breeding is another approach that's quite nice because it, it's based on hypothesis, not just on trial and error. And in rice, we have some nice examples of ideotype breeding. I may not have the time to discuss them here, but what's nice about the ideotype approach is that it's not so dependent on the size of the program, but it would be the smart approach compared to the strong approach of the big programs. The risk is that the breeder can become too passionate about the hypothesis and want to prove it, anyway. And so we should never be passionate about a scientific hypothesis, or else you can make a type 1 error, right? The rice new plant type from Erie is a case of ideotypic hypothesis that didn't work quite the way they meant in the beginning. Okay, but let's discuss this in another occasion. Population breeding has been already discussed here, and the simplest way to understand it is just to increase the frequency of the favorable alleles. But it's incredible the amount of theory, mathematical theory, that has been developed in the 20th century about the quantitative trait breeding in the population setting. So what the take-home message here from population breeding and quantitative genetics is that you consider that you have an infinite amount of genes controlling that trait. So it's like, finely granuled situation where you have an infinite number of genes with an infinitesimal influence on the trait. It's a fluidic way to look at this. And recurrent selection is a system where you go selecting and recombining, and normally, so the mean of your population will moving to the upper side, and this keeps going. Actually, there are some examples of many generations of continuous gains. And the way you could increase the efficiency of this would be through being quicker, being larger, or being more precise. If you increase precision of phenotyping, actually you are increasing the heritability of the trait. That's more genetical and less environmental. And some disturbing forces will create some difficulties for you, like migration, seed contamination, genetic drift, population structure, unintentional selection. For example, the birds can come and eat all of your early plants that headed earlier. So Okay, hybrid breeding. Hybrid breeding is the leading method for cross-pollinating species, especially maize. And here then we reached the peak of the homogeneity paradigm. And here we are with those extremely homogeneous crops or varieties that are saying hybrid cultivars. And it's very business friendly, of course, because you keep control of what you have. And the conclusion of part 2 so is that conventional plant breeding works within the limits of sexual compatibility of closely related species. All right, we normally do everything by crossing. The proposition is that we are still far from reach the limit of this system. We can keep doing genetic progress through those conventional approaches for many years to come. And one question that's a little beyond the scope of this meeting, but I feel that we are putting exceedingly high expectations on plant breeding to solve global sustainability issues. Now, we may be advocating in our favor, trying to draw more funds for breeding or something, but in fact, I think that the expectation is a little too high. And the third part, I'm running out of time, but that would be the part where we know what's going on within the genomes. So there are different ways that you can learn about the relationship between your genome and the trait that you are seeing. You have to build linkage maps, you can do QTL mapping. Those chromosome segment substitution lines are very nice, where you take an exotic germplasm and you introduce small pieces of it into the background of an elite variety. Then you can see the influence of each one of those small fragments, the yellow fragments, in your well-known elite variety. And the association panel, the association method that has been already well covered. Applications of molecular tools in plant breeding, there are many, and this list doesn't cover everything, but you could use it for assessing genetic diversity of parents, accelerating introgression of single genes into elite lines, pyramiding disease resistance genes, selecting genes for controlling traits for difficult traits that are difficult to phenotype, estimating breeding value, fingerprinting released varieties for intellectual property protection, controlling seed quality, so everything from the most scientific applications until the more trivial and commercial applications downstream. You could use, there are different options, technological options. I think that we can jump over this. One of the major applications is exactly to rescue useful genes from exotic or non-adapted germplasm, like this example of Oryza glumaepatula, which is a wild species of rice from South America. You can introduce a small proportion of genes into the Oryza sativa background, but still, in the left corner, that small slice would still be a few thousand genes, proportionally. So there may be unintended consequences. But using markers, you can be more specific and detect the recombinations that would get rid of most of the junk and then bringing into the elite germplasm only the happy allele that you want there to rescue, that has been left behind in the domestication or in the conventional breeding line. And the genomic selection, which is very trendy now, it's interesting because it's the reconciliation between the molecular work and the quantitative genetics approach. Here we are like going back to that infinitesimal control of the trait. You don't really care how many traits, how many genes are controlling the trait here, because you are analyzing thousands of molecular markers and you are using all of them to estimate the breeding value of the individual. So it's like, you know, going back to mid-20th century now with the molecular information in your hand. But it really requires an efficient pipeline for sample handling and data analysis. And this is the last slide, just the conclusion of this third part is that new molecular tools allow better use of allele diversity, making easier handling specific genes and using exotic germplasm. I think all of us could agree on this. The proposition is that molecular breeding will make a large impact on released varieties in the next decades. We're just in the beginning of this. So much more is to come. And the question is, will we ever have the full control on genetic recombination? Not only monitoring it, we will be able to control it in such a way that we will be able to design and build entire genotypes in silico, and then build this plant as an organism that would be much better than, or something that probabilistically you would have to test billions of plants to find that genotype. You may be able to build that from a model based on the knowledge of the effect of thousands and thousands of genes. So that's what I prepared for you. Time runs very fast once you are here. So I'm sorry for running beyond the time. Thank you.
Just one question then. What are examples of traits that have been bred into rice from exotic germplasm?
Well, especially disease resistance genes, they are the typical case. Also, those that new plant type hypothesis, the ideotypic approach also normally takes that one go to a broader germplasm, so they had to use some unadapted varieties, and that was probably where there was a yield penalty that actually was unexpected.
Any other questions? All right, Flavio, thank you so much.